Hello friends, hello students. Today we will learn about gene mapping methods. What are the methods in gene mapping? First is linkage map. Linkage maps using crossing over. Morgan Stu, Sturtevant. Morgan students named Sturtevant that postulated that the greater the distance between the linkage genes, the greater the chance that non-sister chromatids would cross over in the region between the genes, and hence the greater the proportion of recombinants that would be produced. Thus, by determining the frequency of recombinants, we can obtain a measure of the map distance between the genes. One genetic map unit. What do you mean by one genetic map unit? One genetic map unit is defined as the distance between the genes for which one product of meiosis in 100 is recombinant. This is equal to the recombinant frequency of 1% or 0.01. A map unit is sometimes referred to as a centimorgan, cm in short cm, in honor of T.H. Morgan. The recombinant frequency can get a maximum value of 50%, as the rest will be parental combination. Linkage maps or genetic maps are constructed on the basis of the recombination frequency. This method can be used to make genetic linkage maps for Drosophila, maize, mouse, and so on, in which we can observe the recombinant frequency. Genes have been found to occur in a distant set of linkage group, with each linkage group corresponding to one of the pairs of homologous chromosomes in the genome of that species. For example, mouse has 20 pairs of chromosomes, therefore 20 linkage groups. All the linked genes in a linkage group are linearly arranged. Let's know about two factors crosses. If we take two genes at a time and observe its recombinant frequency, then it is termed two-factor cross. If a cross yield a recombinant frequency of 9% between gene A and gene B, then the distance between the two will be 9 map units. Similarly, if another cross yields recombinant frequency of 4% between gene B and gene C, then the distance between the two will be 4 map units. One assumption here is that distance between gene A and gene C will be addition of the above distance. However, this assumption do not hold true with increased distances. It has been observed that when the distance between two genes are large, the recombinant frequency is significantly lower than what additive principle would predict. The reason is that when two loci are far apart, double cross occur at significant frequencies. Now let's know about three factors crosses. These involve three pairs of alleles. They allow us to detect some of the double crossovers which were not recognizable in two factors crosses. In addition, three-factor crosses allow to order the involved pairs of alleles. Now let's know about interference. The detection of double crossing over makes it possible to determine whether exchange in two different regions of a pair of chromosomes are formed independently of each other. If recombination is independent in the two regions, which means that the formation of one crossing over does not alter the probability of second crossing over. The probability of a single recombination of both regions is the product of these separate probabilities. Such deficiencies in the observed number of double recombinants are common. They reflect a phenomenon called chromosomal interference, in which a crossover in one region of a chromosome reduces the probability of a second crossover in a nearby region. Over short genetic distance, chromosome interference is nearly complete. The degree of interference is measured by the coefficient of coincidence, which is simply the ratio of the observed to the accepted frequency of double crossover gametes. That is, coefficient of coincidence equals to observed double crossover frequency divided by expected double crossover frequency, where the expected double crossover frequency is that the predicted if the two crossover are independent events. In the absence of interference, the coefficient of coincidence will equal 1. Sometimes labels of interference are given as the coefficient of interference, which equal 1 minus the coefficient of coincidence. The coefficient of interference will thus be 0 in the absence of interference. Interference value lies between 0 and 1 indicate that positive interference has occurred. Positive interference indicates that one crossover interferes with the occurrence of a second crossover nearby. In certain microorganisms, particularly bacteriophages, coefficient of coincidence greater than 1 often occur. Coefficient of coincidence larger than 1 indicate that the occurrence of one crossover increases the likelihood of additional crossover occurring nearby. This second type of interference is called negative interference. The effect of interference on the relationship between genetic map distance and the frequency of recombination is illustrated as each call is an example of a mapping function, which is the mathematical relation between the genetic across an interval in map units and the observed frequency of recombination across the interval. In other words, a mapping function tells one how to convert a map distance between genetic markers into a recombination frequency between the markers. As we have seen, when the map distance between the markers is small, the recombination frequency equals the map distance. Let's know about tetrad analysis. Tetrad analysis. What is tetrad analysis? Let's know about details about tetrad analysis. In some organisms, 4 haploid cells formed when a mother cell goes through meiosis are held together as a group of 4 cells called a tetrad. The knowledge that a specific group of 4 cells are the products of a single meiosis can be used to estimate the map distance between loci without the use of a mapping function by a procedure called tetrad analysis. Tetrad analysis has been extensively utilized in the fungi such as yeast and neurospora. However, there are some animals, higher plants and algae in which tetrad analysis can be performed. In some organisms, such as Neurospora, the haploid products of meiosis are ordered such that one knows which ones separated at the first meiotic division and which ones separated at the second meiotic division. The extra information can be used to map the location of the centromeres. When a diploid yeast, let's assume Saccharomyces cerevisiae, cell goes through meiosis, the four haploid spores that are formed are held together in an ascus. Ascus means a tiny sac. We characterize tetrads based upon the number of parental and recombinant spores they contain. Consider a cross between an mating type haploid yeast strain of genotype HIS4 TRIP1 with an amating type haploid yeast strain of the opposite genotype HIS4 TRIP1. These two genes are unlinked. The possible kinds of tetrad are parental ditype PD, non-parental ditype NPD, parental ditype, a tetrad containing four haploid cells of the parental classes, non-parental ditype NPD, a tetrad that is a tetrad containing four recombinant haploid cells. The two parental classes have recombined to form the reciprocal non-parental recombination of alleles. Now let's know about tetratype. In short, T. A tetrad containing four kinds of haploid cells, two different parental class spores and two different recombinant class spores. In crosses involving two unlinked genes, tetratypes arise when a crossover occurs between one of the two genes and its centromeres. If let's know about some points about PD and NPD. If PD is equal to NPD, then the two genes are unlinked. Either they are located on different chromosomes or they are far apart on the same chromosome. And then second point is, if PD is greater than NPD, then the two genes are linked. When PD is greater than NPD, when PD is smaller than NPD, the haploid spores of the parental class significantly outnumber the haploid of the recombinant class, a sign of linkage. Let's take an example so that you can understand this concept. Parents. Suppose we take two parents, ARG3 URA2 cross ARG3 URA2. First one is smaller, second one is greater, in capital letter. Okay. Diploid, diploid is small ARG3 small URA2 by capital ARG3 capital URA2. Now let's know about meiotic products. PD. Here PD is ARG3 URA2. These are small ARG3 small URA2 small, comma capital ARG3 URA2 capital, again ARG3 URA2 close bracket close equals to 127. Okay, it comes in number 127. Now come to NPD. ARG3 small ARG3 URA capital URA2 small arg 3, capital URA 2, capital ARG 3, small URA 2, capital ARG 3, small URA 2. It's equals to 3. Now come to T. T it's a small ARG 3, small URA 2, comma a small ARG 3, small capital URA 3, URA 2, comma capital ARG 3, small URA 2, capital ARG 3, capital URA 2 equals to total is 70. Okay. Now recombinant frequency is RF equals to recombinant frequency in short RF equals to NPD plus half T divided by total number of tetrads into 100. So here in this example 3 plus 1 by 2 into 7 divided by 200 into 100 equals to total is 90 map units. The equation will be modified to be obtain a better estimate of map distance. Understood? Now let's know about ordered tetrads and gene centromere distance. The Neurospora crassa, meiosis occurs. Here meiosis occurs within the tight confines of a narrow ascus, resulting in the formation of ordered tetrads. Because of the precise positions of each meiotic product within the ascus, one can interfere the arrangement of each chromatid of homologous chromosomes during meiosis 1 and meiosis 2. This gives information about the distance between the gene and its centromere. Meiosis 2 is followed by mitosis. Each pair of genetically identical daughters sites adjacent to one another. Each ascus is thus made up of 8 haploid ascospores. We will take an example in which we will look at the outcomes when there is and is not a crossover between a locus A and the centromere. When there is a no crossover, the four A spores are at one end of the ascus and the 4A spore at the other end. The different alleles capital A, small a segregated apart at the first meiotic division. This pattern capital A is to capital A is to capital A is to capital A is to small 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 a. For capital A is to 4 small a is called first division segregates, that is in short FD, first division segregation. The lower panel of the lower panel when there is a crossover between the locus and its centromere, meiosis 1 produces 2 daughter cells, each of which is capital A, small a. The different allele capital A and small a then segregate apart at the second meiotic division. After meiotic division, the final pattern is capital A is to capital A is to small a is to small a is to capital A is to capital A is to small a is to small a, which is called second division segregation. Since ASCII showing SD have 2 chromosomes with recombination between the centromere and the locus plus 2 non-recombinant chromosomes, we can calculate the distance between the locus and the centromere as D equals to 1 by 2, that is small d equals to 1 by 2 whole of SD divided by N bracket close divided by into 100. Again I am repeating, small d equals to half of SD divided by N whole into 100. There is a 1 by 2 in the equation. Only 1 by 2 of the chromosomes in a SD ascus are recombinant. There are a total of 300 ASCII, of which 100 are SD. So, small d equals to 1 by 2, 100 divided by 300 into 100 equals to 16.6. 16.6 MU, that means map units. 16.6 map units. As the distance increases up to 50 units, in theory, all ASCII should result from second division segregation. However, numerous factors prevent this. As in diploid organisms, accuracy is greatest when the gene and the chromosomes are relatively close together. Mapping with molecular markers. The molecular markers is a site heterozygosity for some type of silent DNA variation not associated with any measurable phenotypic variation. Such a DNA locus, when heterozygous, can be used in mapping analysis. Just a conventional heterozygous alleles pair can be used. The three most common type of markers used today are RFLP, RAPD, isozyme. RFLP is restriction fragment length polymorphism. RAPD is random amplified polymorphic DNA. Isozyme. These three RFLP is mostly used. RFLP markers have several advantages in comparison with the RAPD and isozyme markers. They are codominant and unaffected by the environment. Any source DNA can be used for the analysis. Many markers can be mapped in a population that is not stressed by the effects of phenotypic mutation. The primary drawback to RAPD markers is that they are dominant and therefore heterozygous individuals cannot be distinguished. They are codominant and unaffected by the environment. Any source DNA can be used for the analysis. Many markers can be mapped in a population that is not stressed by the effects of phenotypic mutations. The primary drawback to RAPD markers is that they are dominant and therefore heterozygous individuals cannot be distinguished. The weakness of isozyme markers is that each of the proteins that are being scored may not be expressed in the same tissue and at the same time in development. Therefore, several samplings of genetic population need to be made. RFLP, restriction fragment length polymorphism, a molecular marker based on the differential hybridization of cloned DNA to DNA fragments in a simple restriction enzyme digested DNAs. Now let's know about RFLP, restriction fragment length polymorphism, a molecular marker based on the differential hybridization of cloned DNA to DNA fragments in a simple restriction enzyme digested DNAs. The marker is specific to a single clone or restriction enzyme combination. Now let's know about RAPD, randomly amplified polymorphic DNA, a molecular based on the differential PCR amplification of a sample of DNA from short oligonucleotide sequences. Now let's know about isozyme, a molecular markers system based on the staining of proteins with identical function but different electrophoretic mobilities. Let's know about mapping by using somatic cell hybrids. The linkage groups of humans cannot be investigated by designed crosses used with Other species for this other methodologies like somatic cell hybridization and chromosome banding techniques are used. Mouse human somatic cell hybrids have been used for mapping human genes. Somatic cell hybridization was first demonstrated with mouse cells by G. Bursky and colleagues in 1960. Somatic cell hybrids are culture lines that contain the entire complement of the mouse genome and a few human chromosomes. These culture lines are developed by mixing human and mouse cells in the presence of the ultraviolet inactivated Sendai virus or a chemical polyethylene glycol PEG. Any one of them facilitate the fusing of the two cell type to form a hybrid cells. Initially, the nuclei of two cells are separated. This stage is called heterokaryon. When both the nuclei fuse together, then it is fused. It is referred as synkaryon. For a reason that is not entirely known, most but not all human chromosomes are lost from the hybrid cell lines. Usually, a few human chromosomes are retained. Because the human and most mouse chromosomes can be distinguished by chromosome staining techniques, it can be determined which human cells are retained with a specific cell line. Any questions? Now completed gene mapping. Any questions regarding this gene mapping? You can ask me in the comment box. I will clear all the doubts. Thank you.